Good afternoon, everyone. My name is Beth Foss, and I am with the Choroideremia Research Foundation. And we're going to continue in our science series and learn about a great new program, a clinical trial series that Ray Therapeutics has launched. And we have two wonderful people joining us today. We have Rebecca Kramer is a low vision specialist, and she'll be starting us out, and then we'll take a couple questions, followed by Dr. Raj, I want to make sure I get this correctly, Raj Agrawal, and he is the Clinical Development Director at Ray Therapeutics. We're really excited to have you both today. Thank you so much for coming. And go ahead and, as we said, folks, typically what we do is use both the chat feature or the Q&A, and I will be running that, and I will pose your questions to the two of these wonderful individuals. And let's get started. Thank you so much again for joining.
Good morning, everybody. Afternoon, evening, wherever you are. I'm in California. I am an optometrist by training, low vision optometry, and then later got a PhD as well. And I got into clinical trial research for inherited retinal diseases about 10 years ago or so, working with stem cell therapy program for retinitis pigmentosa, and then consulting on a few other projects with small interventions, or whether they were cell therapies or even medical device. But really my specialty is working with patients with visual impairment, and it's very needed when we talk about studies and how to actually measure if vision that is changing or if these treatments are effective. Now, maybe they're effective for slowing the progression of a disease, or maybe they're effective for actually improving vision by a certain amount, but what does that amount look like? What tests do we use, and so on. So I have a special heart for that. And so I'm here today to kind of collaborate with Ray Therapeutics on their program and what they're working on in their studies. And so we'll kind of share a little bit of what they're doing, but also from my background with other programs and other research. Endpoints, or the assessments and tests that are used to determine whether something is effective. I'm going to focus today on things affecting vision. So we're not really imaging and not really anatomical structures and so on. I know you've had some great talks in the past, even in the recent past, with scientists talking about maybe multiple areas of testing that's used to show whether a treatment is effective. But I'm going to really stick on vision. How do we measure vision? And then functional vision, how it affects life. Are there certain tests and so on we can show that a treatment is actually moving the meter in terms of mattering to patients? And in this area, one of the main things when we're working with regulatory groups, especially in the United States, the FDA, you have to come with tests that they agree are showing this change. And there's been some long historical tests that they agree that, hey, this works well as a test. Some haven't changed in many, many, many years. And visual acuity is one of those, the letter charts and how far down on that letter chart that you can read. And they've established pretty well clear that 3 lines of vision gain is meaningful, meaning if we go from the top line of the chart to about midway in the chart, as you're reading those letters down there, that is beyond kind of day-to-day variability, and that is beyond maybe just a little bit of fluctuation, but really would be meaningful to patients. And that's been demonstrated over the years as to why that's true. But if you are someone with vision who you can't see the letter chart anymore, then that's not a very useful test, and it's not a very useful program if you're working on some type of intervention, or there's a study that's measuring vision in people with close to light perception vision, or maybe no vision at all. But today I'm talking more about there's a little bit of vision there. How do we measure that? And so for CHM specifically, we've got to talk about what tests or assessment are useful for that particular condition, because something that I might use for macular degeneration, where I'm missing central vision, but I have good peripheral vision, is not going to be useful for a program that's working with CHM, where we've got peripheral vision loss and we've got a very central small field that maybe actually isn't impacted by vision loss or visual acuity specifically for a long time in the disease. So when we're considering what kind of endpoints, it's really important to say, who's developing this for a company, who's working on the project. Are they familiar with the disease and the vision science around the tests that are out there already? And the approach that company is taking for the treatment, is it accepted by FDA already, or does it need some development work and then it needs to go to the FDA to have some dialogue and collaboration around that. I found that the FDA is very collaborative and actually does want to develop, or for groups to develop these, but they're not necessarily going to develop their own endpoints and put it out there. It's going to come from the industry, from companies, from scientists who are proposing with good science what tests should be used in what program. And one size does not fit all. You don't just use one test for one disease and so on. So 3 key areas I'm going to focus on this morning for me, is the treatment mechanism. How does the mechanism of action? What is the treatment actually doing? What is it proposed to do? What is it done in animal studies and so on, if it's first time in humans? What do we kind of expect in terms of the impact? Is it going to impact, is it supposed to impact the visual field and maybe open things up? Is it going to impact one little spot way off on the side somewhere? Or is it going to impact, theoretically anyway, central vision? So we want to look at how the treatment itself is aimed, and then the disease indication. So is it RP? Is it retinitis pigmentosa? Is it choroideremia? Is it Stargardt? Is it macular degeneration? That's really important because one test, again, is probably not going to be useful across all these types of diseases. And then a really important part is the disease stage. Is it the beginning of the disease? Is it the end stage of the disease? And how are those things characterized? And some diseases are very well laid out in the scientific literature and course of things, but others maybe don't have as much work done for them, especially more rare diseases. So let's take a look just real quickly at treatment mechanism that are out there in some programs and so on. Just generally, and I won't name names of who's doing what, but we have gene therapies, we've got stem cell therapies. Optogenetics is such an interesting field, and Raj will talk a little bit more about that. Dr. Agarwal will talk about that. I'll just briefly mention its kind of mechanism of action as it's relevant to this talk. And then even retinal prosthesis, maybe something's been implanted and it's bypassing the retina altogether. So each of those has a functional way that it's trying to either restore vision or slow the progression of the disease. And then ultimately that has an impact on the patient, of course. So how do we measure that? So it's important we take into account that treatment mechanism. So for optogenetics, as a very kind of unique gene therapy type of approach, it really is creating light sensitivities in a different layer of the retina besides your photoreceptors that are so typical. So this is neat because we can have photoreceptor loss, so a later stage of the disease ultimately, and still be able to, let's say, rescue some light ability by changing the light sensitivity of retinal ganglion cells or bipolar cells. And those have a different structure than photoreceptors. So we need to use the science that we know about how things are laid out, how many cells are in the retina. So if I have an intervention to retinal ganglion cells or bipolar cells, is it going to be across the whole area? And this may depend on how it's placed in the eye. So like Ray Therapeutics is using an intravitreal injection. So if this is effective and if it actually makes it to its place that it's supposed to, it could be a large area that's treated in the retina and maybe not just the tiny 2 degrees or 3 degrees that's in the fovea. A lot of programs are working, have done subretinal injections. And so those might only reach a small area where the actual treatment area has been placed under the retina. And so that will impact what kind of tests are we going to measure those areas with. So now let's talk a little bit about the disease indication, choroideremia specifically. And we know some things about this. Of course, many of you are living this experience with a constricted visual field, decreased vision, maybe centrally as the disease progresses. Sometimes you have good pinpoint vision till late in the disease. So knowing what stage we're talking about. The contrast sensitivity, your ability to see light grays and shades of gray, that's impacted pretty early on. So that's an important metric all the way across the early to late stage of the disease. Glare sensitivity, color vision, those things are going to be impacted. And then when we look at how that impacts the person, which is really about kind of functional vision, the slide I'm showing now is just, it's a diagram. It's a little busy, but for a clinician, it's a really nice way to say, look, the person really matters here. It's not just about treating the eye itself or even restoring some visual acuity. How does that actually impact the person? And then society, am I participating in life? Am I able to work? Am I able to age in place and so on? So the things related to kind of the person functional, that might be about mobility. So maybe we have a test that measures mobility or something else in a daily life of being able to see your medications or manage your own medications. And those kind of things would be more functional vision. And let's spend a minute on disease stage. So some of these programs, again, some of these great talks that you guys have already had, they've really been, at least when I listened to them, I was super impressed, but they were, even from a talk in 2022 with a scientist, I'm going to mess up her name, I'm sure, but from the UK, it was a little more about earlier stage disease, or when we're looking at how some of these earlier functions are impacted. So microperimetry or some of these visual field tests might be useful for an earlier stage patient. So when we're looking at optogenetics, though, it's going to be a later stage because we can bypass the photoreceptors. So we can go into, we don't have to have them in good order. So let's talk a little bit more about that then. So if we say that we're looking at optogenetics, we're looking at choroideremia later stage, we know visual acuity may be impacted. And so if we're talking about Ray Therapeutics and we're talking about visual acuity, it is the people that are in their studies are going to have visual acuity impacted. So, and visual field is going to be more constricted, maybe islands of vision and so on. Contrast sensitivity will definitely be impacted in these patients in the stage of disease for the studies. And night blindness, how you function in low light, is a key thing. So we think about that, think about who the patient is. And we say, well, if visual acuity is impacted, or we're including people in the study who have poor central vision, even if it's a really small or a large field of area, how do we measure that? If we can't get it on the letter chart, which we'll try with some patients, but then what if you can't get any letters? Well, that's where grading acuity might come in. So using black and white stripes in order to detect different sizes of those stripes on a screen will be a little easier of a task because you don't have to identify what letter if you have a very small visual field area. So gradings is a nice way to do that. Well, we need to make an agreement with the FDA around how much change in the grading vision changes. You might not be able to just apply the same things that have been accepted about 3 lines or a certain change on the eye chart, jumping over to gradings and having the same type of measures. So we have to reach some agreement around that. Other areas that we might be using for this later stage condition is contrast sensitivity, if we can measure it. Now, traditional approaches are using letter charts. So again, what if we know how much the FDA generally accepts for change for that. So what we always want to do when we take a test is say, are the tests that are out there that are already being used, can they be used here? Because it's much better to take something that it has a track record and is accepted, it's easier, faster to get to your end goal. But if they don't adequately measure the level of vision that you're working on in that particular program or intervention, then that doesn't match up, and you might have a treatment that never makes it to market because the wrong tests were used to show this change. So visual fields, again, we can say, well, let's measure visual field, but if you don't have someone who can actually fixate on a tiny dot in this machine that you're typically using for visual field, then you can't use the traditional fields like kinetic visual fields or even any other static type of visual field measures if you can't hold fixation. So what do we do there? And then in night low vision, we've got some tests such as mobility tests with seeing arrows on the ground. But what if your vision's too poor and you can't see arrows on the ground? But you can detect some high contrast edges. Maybe we can use that for a version of some type of test. So when I teach others or we talk about what are the steps that you want to go through as a company to either develop or utilize tests that are out there, we want to say, can we modify an endpoint that's already out there? Do we have to create it from scratch? If we have to create it from scratch, we never have to do anything from scratch. There's great, brilliant minds out there have done something. Take that, and then how do we validate it? How do we modify it for a particular patient population in our program? And how do we then show the science that it's a good test and it isn't going to change without us kind of knowing how much? Like, you're not going to come in, test one day and test the next day and have totally different results. That wouldn't be a very good test. And then collaboration, who do you collaborate with to develop those tests? And is there rigor in the science that you're creating those endpoints? Sometimes it could take several years to develop a test and get all the science that you need to support it. So doing that in parallel with your development program is key. So you're not doing one after the other and taking 20 years to develop a treatment. Things can be done in parallel, and they can be done fairly quickly if you have the right team, the right partners, and so on. I use the word qualifying an endpoint for if you have one that's pretty close, but now you want to check it in. It's been used in another program, but you want to check it in your particular population. So you don't have to do as many tests and as many validation studies, but you might need a study just to show that this test does work in your population. How much development is needed kind of depends where you're starting and how much track record they have already with the agency and in publications. So, picking the partner that you work with. Ray Therapeutics has worked with a group called Adaptive Sensory Technology. They already have a kind of a letter type of chart for a letter test for contrast sensitivity. And now they're developing a grading test. Well, again, those gradings are these stripes. And so if we can show those stripes not only in high contrast black and white when we're measuring visual acuity or the precision of vision, now if we can change those gradings to have a gray color to them, different shades of gray, we can see if someone has some level of contrast sensitivity versus maybe none at all. And maybe the treatment really improves and increases it. So those validation activities to take a test and then, go through the process of the rigor with the science and so on. There's a lot of questions that we have to ask. And there's been some pathways that have been demonstrated by the FDA and with just good vision scientists about, you got to show some retest reliability. You got to, who's interpreting the test. We have to determine how much change is really clinically meaningful. And there's ways to do that. Not to oversimplify it, but there's a process there. So I'm not going to go into all of those steps, just that those steps are usually needed. This slide is just an example of a test on day 1 and then a test on day 2. A little different response on each, but very similar in terms of the score. And that score is the important thing to say, okay, if we know, I'm having a bad vision day and I'm having a good vision day, that happens a lot. Is the bad vision day a lot different than the good vision day, or is it my perception of it? How do we measure it and say, okay, in this population of later stage choroideremia, we expect a certain amount of fluctuation just in the given day and fatigue and so on, how the mood that I'm in. And then but sometimes you don't get to choose when you're testing. The study time, the study dates require a certain window of time. So we need to make sure that the tests are kind of, if you get some major improvement, it's beyond just kind of that day-to-day fluctuation. I'll spend a couple minutes here just on low luminance mobility tests. This has been a big field since Luxturna had their test. I helped develop a test with J-site, with stem cell therapy, and this was kind of arrows on the ground, and it kind of expanded upon the work that Spark and Luxturna and those good scientists had done. But you say, well, okay, that's nice to take something that's been done, but if it doesn't work in our patient group, what do we do? So here's a test that Ray Therapeutics has started working on, is a kind of a wayfinding is what we can call it. And there's been a couple of publications. The clips on the top of the slide are just not to be read. They're just to show there's a couple of publications from groups that have been working in this area the last few years. And really just kind of navigating major, hallways and being able to identify landmarks for navigation. And so this particular one is a pathway with just black and white stripes. Kind of staying on that pathway is the goal of the test. Nothing too complicated, no obstacles. I mean, think about who this test might be for. Would be someone with pretty small central vision, pretty blurred vision, so not very good resolution. And even in the highest lighting levels, maybe could pass that, walk from one end to the other of the room. And then once we drop into lower light levels, can't do it at all. So now maybe the treatment and the intervention shows that, well, not only can they do it in high lighting, but they can also do it in really pretty dim lighting. That would be pretty exciting to show change. But, maybe depending on the treatment, we need to tweak the test a little bit or change it and say, what, we're not going to deal with the low light. We're really happy with any changes in high illumination. So let's just measure it in high illumination. And then we figure out the scoring and talk to the FDA about how to manage that. One thing that's important in all of these tests is to, make sure that the patient, the person who's doing the test, who's undergoing the study, that they're relaxed, that they're reassured, that they're encouraged. It's not in a really busy clinic setting where it's kind of rushing from one thing to the next. That can cause so much anxiety, and all of those factors can impact how someone performs on the test. So I usually recommend to groups that I, consult for, or if it's just asking in a passing thing, if you can set up a special clinic or a space for this testing. It takes many hours to kind of give time to go through various trials, give breaks, coffee breaks, tea breaks, and things like that. Make sure you have enough food, and then assess the patient as they're going through. If it's been 3 hours and they're just exhausted and everything's starting to become variable, maybe we need to split that testing into 2 days. Some of that flexibility is just too hard in a large ophthalmology clinic. And so being able to outsource that or have it offsite somewhere has been very powerful in the studies that I've worked on. So this can help you then be creative if it's somewhere offsite to be able to create more tests or to bring in something that might be relevant as the program is going along and as the group is discovering more about their treatment. I won't spend too much time on these other. I think we're about ready to wrap into kind of question time. But let me just see if there's any other key things here. Well, here's an example of a test that was published in 2021. Dr. Sahel, who was working with patients, and this was in an optogenetic therapy program. It's just highlighting one patient, but they had creatively developed a test just with cups and black against white cups, and then even varying the color of the cups. And so patients had identified, was there even cups on the table, how many cups, and what location. So you could actually get a little more complicated with this task if someone could just identify that there was or wasn't a cup. That could be like a 50-50 chance of them getting it right. It's not a very great discriminator for a test, but once we get into maybe 3 or 4 positions and they have to correctly identify the position of the cup, now you've got more than chance. And so that's the makings of a good test. So this is one that could be used. And then when you take a test like this that's been published, it's exciting because now you can say, okay, there's a foundation. How can I tweak it, change it a little bit, and make it more relevant to my particular program? And that's what companies should be able to have the flexibility and the scientific team to work on. So I will stop there and take any questions, but hopefully I've gone through a little overview of why the tests that you pick are really important, ability to modify those tests, but matching those tests up with the stage of the disease, the type of the disease, and the treatment pathway, how it's actually affecting cells, what kind of vision changes might we expect in a program.
That was great. Thank you. Thank you very much. We do have 3 questions that are here. I think I'll just pose these now. And maybe, Raj, if you want to take them or later, we can discuss that then. The first question is, for CHM patients who have participated in prior gene therapy clinical trials, such as Nightstar, on one eye in the operation room, not the control group, would they still be able to be qualified for this new trial on the same eye?
So hi, everyone. This is Raj. I guess this question is for me. I'm going to be discussing this as part of my talk later on. But one of the exclusion criteria we do have as part of the protocol is a patient who has been previously a part of another gene therapy study. So most likely this patient will probably not be eligible for this study. But we can discuss more, yeah.
Okay. 2 more questions. For many eye diseases like CHM, stopping further progression is already a great gain. Good point. Many endpoints are about visual field, letter charts, or another measurement of improvement. How about measurement of stabilization of sight? During yearly checkups, we do eye scans. If you would follow a patient 10 years, for instance, and in one eye there is no change, the treated eye, and one eye untreated, there is change, that would be a good endpoint. Excuse me. It emphasizes firstly about stopping further progression. Restoring of eyesight is for many a later second step. Can you talk to this?
Becky, you want to go ahead?
Yeah, I can start, and then you can take over as a retina specialist. You've got a lot of good insight in this. For me, yes, it's a good description what you just described about tracking it over a long period of time. The challenge with these tests, any test of vision, is that you have some variation from day to day. So you have to have enough progression in the bad eye and to compare it to the treated eye to show there really is a difference. And that takes many years. Some people, the progression of the disease is maybe one bad year and there's a drop off, but most times it's a slower progression. So that means that the studies might need to be 5 years long in order to get beyond kind of showing that, oh, that's just normal hope or placebo effect. And that's a challenge for companies because now they've got to go on a really long ride to slow progression. And it's very expensive to do these trials. So that's my, it's so important, but it's hard to do.
Did you want to add anything to that, Raj?
Yeah, I'll just add something to it. So, and that's a great question, by the way. And what Becky mentioned in regards to, or Dr. Cameron mentioned in regards to, the fact that we require a long enough trial to showcase a benefit in terms of, vision not worsening. 3 things I wanted to point out in addition to what she mentioned. One is, we do have enough natural history data of study of disease progression in choroideremia. So we do know how the vision progresses over time. So that could factor in conversations with especially with the regulatory authorities to figure out what is a possible way of trying to figure out an endpoint for the study. Um, it's important for us as Dr. Cameron mentioned in our talk previously, it is important for us to have conversations with the regulatory agencies to figure out these endpoints. And as of now, majority of the trials require us to show 3 lines of improvement, and that is really difficult in some of these inherited retinal degenerative cases. And we all know from the study, the phase 3 study, the STAR study that was done, patients did not meet that primary endpoint of 3 lines of improvement. But there were patients who had 2 lines of improvement in that study. So if the endpoint would have been 2 lines of improvement, potentially that study could have crossed the hurdle of getting an approval. So it requires a lot of conversation, like I said, with the regulatory authorities. But agencies have been open to suggestions. So just to give an example, completely unrelated to this, diabetic retinopathy as a disease has, obviously has similar line in terms of slow progression. FDA has now agreed to showcase the fact as an endpoint of stability of the disease rather than looking at 3 lines of improvement, especially for diabetic retinopathy. So there is some room in terms of having these conversations and getting some feedback from the FDA. And hopefully in time to come, we should be able to get something going.
Let's take one more question and then we'll let you get started. Someone writes in, I was just having this conversation with my retinal specialist. Get my bifocals here. Tunnel vision has led me to losing my ability to track and find things. A standard vision test makes me seem way better on paper than I am in real life. Are there any tests to gauge this better for us CHMers?
Yeah, I don't know when that question was logged. Was it at the beginning of the talk?
About midpoint.
Okay. Well, I got a little bit more into the functional test. So yes, I totally agree that, visual acuity is just measuring your letter size does not represent the challenges of the disease, especially when field has gone quite low, or it's the pattern, or just contrast sensitivity, which is ability to detect shades of gray. So no one test does all of that. And I think that's where we need to have a battery of tests, several tests that are describing vision, and especially ones where, again, the treatment is aimed at the most for improving. That's the way it's going to be. And I think the FDA appreciates that. They want multiple tests that kind of lean the same way, so you're not all counting on just one test. But yeah, that's where my whole talk about functional vision comes in, and we need a few more developed. But there are a lot of creative scientists out there that are working on these types of functional tests. Sometimes a mobility test isn't the ideal one, but these efforts are really important for the field.
Certainly. All right, well, let's just hold the rest of them. And Raj, if you would like to go ahead and start, I'll turn off my camera.
All right. So thanks, everyone. I'm just going to move my slides. Okay. So good morning, good afternoon, everyone. My name is Raj Agrawal, and I'm the Vice President for Clinical Development at Ray Therapeutics. I would take this opportunity on behalf of the company to thank Kathy and Beth and others at Choroideremia Research Foundation for this opportunity to present our work to you all. And I also obviously appreciate all of you joining in this conversation. So thank you. Just to mention, I joined Ray Therapeutics late last year, and I'm really excited working on this technology and the clinical study that we have ongoing at this time. Hopefully this study progresses and reaches all the way to approval. So before I dive deep into the work that we do at Ray Therapeutics, I just want to take a moment to introduce you all to the company. Ray Therapeutics is a clinical stage company headquartered in Berkeley, California, and we also have a center in Irvine, California called the Vision Research and Assessment Institute, which I'll be addressing in some more detail at the end of this talk. The 2 lead assets we have currently include RTX-015, which is currently focused on choroideremia and retinitis pigmentosa, as well as potentially has implications for other IRD diseases, as well as the second product is RTX-021, which is focused on Stargardts and AMD. RTX-021 will be starting a clinical study end of this year. These two bioengineered optogenetic medicines have been optimized for human vision with the aim of restoring higher visual function without the need for specialized goggles, as we have seen with some other attempts. The management team of this company has significant experience in the field of regenerative medicine and optogenetics. As shown in the slide, with RTX-015, we are currently in a phase 1/2 clinical study for retinitis pigmentosa and choroideremia. We are hoping to start enrolling patients for choroideremia very soon. FDA has given us the go-ahead, as well as we've gotten approval from the IRB to start the study. And as I mentioned, RTX-021 will be starting late this year in Stargardt's. So just to give a little agenda of what this talk entails, it will highlight some aspects of the current research in choroideremia, some of which Dr. Cameron has already kind of highlighted in her talk, followed by a deep dive into optogenetics, and ultimately focus on RTX-015 and the clinical study. And lastly, I'm going to provide a little description about the RAY Institute, or the Vision Research and Assessment Institute, that we have in Irvine, California, which is importantly involved in this clinical study that I'm referencing. So a lot of basic and clinical research has led us to where we are today, with ongoing research by various groups around the world to develop a treatment for choroideremia. We had, as I mentioned earlier, as in regards to the answer to the question that was asked about patients eligible and endpoints and all that, we had a significant study that initially reported in 2014, where patient outcomes showed significant benefits actually, in a phase 1/2 study following retinal gene therapy for patients with choroideremia. But unfortunately, when this study was taken into phase 3, it did not meet its primary endpoint and hence did not progress further beyond this. Most of these studies require subretinal injection, which is a complex and challenging surgical process and has the potential to cause intraoperative or even long-term complications related to the procedure itself. When we look at stem cells and retinal implants, they also equally require complex surgical interventions. And again, that can potentially be a cause during the surgery itself, or may have long-term implications in terms of outcomes later on. The last bullet point points out to molecular photoswitch, which is currently being tried out as a potential treatment. So based on what we know so far, it is likely to be a monthly injection, which is likely to be a treatment burden on patients. We all know that getting injections every month is not a pleasant experience. So it is potentially, as I mentioned, a treatment burden on patients if this study continues. Compared to all that, optogenetics does have significant advantages that I will be discussing in the next slide. So what is optogenetics? It is a therapeutic approach that involves using genetic techniques to introduce light-sensitive proteins, such as channel rhodopsins, into retinal cells that are normally not light-sensitive. Once injected, after the cells are transduced, these cells are able to respond to light and restore vision. It involves a viral vector, usually an AAV vector, that is used to introduce genes encoding light-sensitive proteins into specific retinal cells, and in our case, mainly the retinal ganglion cells. After expression of the optogenetic proteins, the treated cells can detect light and are able to send visual signals to the brain, and thus bypassing lost photoreceptors. That obviously happens in this study. So what are the advantages of this technology? The most important and significant aspect is that this method can work even when photoreceptors are completely lost. So definitely a big advantage. It is also less invasive since it requires a simple one-time intravitreal injection. So we believe that just one single injection will potentially be able to help patients in the long term. Importantly, what we have at Ray Therapeutics has shown to work in ambient light, which means the patient will not be required to use specialized biomimetic goggles to be able to see. So again, huge advantages compared to some other attempts that have been made in the field so far. There's a long history of optogenetics behind us. It has about almost 2 decades of innovation and research with pioneering scientists who have worked on this technology since 2003. Since it was discovered then, and with visual optogenetics described in 2005, we have come a long way with the seminal paper that Dr. Sahil and his group published in 2021 about a patient who had received optogenetic treatment and was able to see was able to see much better than what he was before the treatment. That same year, we at Ray Therapeutics bioengineered a channel rhodopsin protein, which has now gone on to be part of the clinical trials since 2023. So just to mention, we stand on the shoulders of giants who pioneered the work in the field of optogenetics. And again, we at Ray Therapeutics are moving this legacy forward with our ongoing work in the field in retinal diseases such as choroideremia and retinitis pigmentosa. Just to highlight what happens in the disease, the cartoon on the slide on the top shows changes in the retina with disease progression, with damage to the retinal pigment epithelium and outer retina, including photoreceptors over time. The inner retinal cells, specifically the retinal ganglion cells and associated, they remain intact and are functional even in late stages of the disease, which is what we utilize for this treatment. While gene-specific gene replacement therapies, mutation-specific gene editing treatments, and neuroprotection or cell therapies are likely to be more useful in early or intermediate stages of the disease, optogenetics is most likely to be useful for patients with late-stage disease, which is again an important factor because of the fact that a significant number of patients do end up in late stage. So just to give a little brief about what kind of work that has been done, and this work is being referenced from Dr. Pan's lab in Wayne State in Ohio. This slide shows the evaluation of RTX-015 in animal models. The study utilized triple knockout transgenic mice, which were chosen for the study because they lack an optometer response, the pupillary constriction, and they have apparent photoreceptor death. The use of this mouse line enables assessment of the restoration of visual function by optometer behavioral assays without the confounding elements that are seen in other animal models. So again, TKO model is definitely a better way to test these treatment models in in preclinical studies before we move on to human studies. The optometry system allows for testing of animal models in a controlled environment with the LCD monitor-based visual system. The test consists of a rotating drum that is centered over the animal's head, which is utilized to measure the optometer response reflex. This reflex involves monitoring of the head movement of the mice in response to visual patterns being presented, typically moving stripes, and is used to gauge visual thresholds for each eye. The optometer response test is an objective, reproducible test that can evaluate various aspects to define improvement in assessments such as visual acuity, light sensitivity, and contrast sensitivity, all of which are important for us in terms of showcasing benefit in choroideremia. In animal studies, RTX-015 has demonstrated 100 to 1,000 times superiority in light sensitivity compared to other attempts. So the graph on the slide showcases the fact that RTX-015, all the way on the top, is shown to be activated in low light compared to the other attempts that are referenced below. And those two dashed lines basically indicate, anything that is below that will potentially require goggles, biomimetic or specialized goggles for the patients to be able to see again. While RTX-015, all the way on the top, beyond that level, does not require any goggles to be used, and it works in ambient light situations. So going over a couple of outcomes in that animal model, let's focus on the graph on the left. This shows visual acuity improvement in animal models that received RTX-015, while the left cohort of animals that were in this study were untreated TKO mice who, as expected, did not demonstrate any improvement. The middle column shows RTX-015 treated mice that showed improvement of 0.22 cycles per degree, which is remarkable when you compare that to 0.40 cycles per degree observed in normally sighted mice. So there's a huge improvement in those animals that received RTX-015. And this improvement was seen to be stable over 1 year after treatment. So again, a big significant improvement and stability and durability of the intervention. The graph that we see in the center was to evaluate contrast sensitivity. Mice treated with RTX-015 exhibited contrast sensitivity of up to 6.7 with an average value of 4.2, which was remarkably good compared to those with normal sighted mice. And again, when you go to the graph on the right, with the optometry motion detection test, the contrast sensitivity functions in RTX-015 treated mice exhibited the peak sensitive speed of 12 degrees per second, which was the same as seen in normally sighted mice, so almost equivalent. So in a sense, with this data, RTX-015 has demonstrated 50% improvement in visual acuity, almost near normal contrast sensitivity and motion detection. So again, significant data outcome from this particular study out of Dr. Pan's lab in Ohio. To review what we presented in all the previous slides, the bioengineered and optimized RTX-015 protein, like I said, shows 100 to 1,000 times better light sensitivity compared to other attempts, while demonstrating improvement in visual acuity, contrast sensitivity, and motion detection in animal models. And it works in ambient lighting. And delivered as a single one-time intravitreal injection, this treatment will avoid surgical challenges and potential complications that come with subretinal injections. Once injected, it will diffuse into the vitreous cavity and transduce a broader swath of retinal ganglion cells, as Dr. Kammer mentioned, compared to subretinal injections, which is usually a localized transduction. Which brings me to the current clinical study. We have this study currently ongoing, which is a phase 1/2 study to look at safety aspects, but also look at efficacy in patients with choroideremia. With a go-ahead, like I said, from the FDA and IRB approval, we are ready to start recruiting patients for the study. Currently, we have 3 sites that are headed by 3 principal investigators. All of them are leading figures in the retinal space. Dr. Barry Cooperman, who's the chief of the Department of Ophthalmology and University of California, Irvine, in Irvine, California. Dr. David Boyer, who's the senior managing partner at the Retina Vitreous Associates in Los Angeles, and Dr. Kenneth Fan, who's an IRD specialist and works at the Retina Consultants of America in Houston. So these 3 PIs are, like I said, significantly known individuals in the space, and we are working with them closely in terms of enrolling patients into the study. This study will be 12 months, and what we are looking for are 2 cohorts of patients, of 3 patients in each cohort, for a total of 6 choroideremia patients. There was a question about eligibility for somebody who's been part of a previous gene therapy study. Unfortunately, again, we can discuss that in more detail, but anyone who's participated in another gene therapy or a cell therapy study or a retinal implant trial, unfortunately, is not likely to be included in this particular study, primarily because this is a phase 1/2 study. We may define the criteria in the phase 3 study later on, but for now, unfortunately, that kind of stays. So in terms of eligibility criteria for inclusion, anyone who's 18 or above is eligible for the study with a confirmed diagnosis of choroideremia. And obviously, this includes a genetic test. If the patient has not undergone a genetic test, we would definitely be able to do that at the site before the patient gets enrolled into the study. In terms of vision, what we're looking for, again, as defined in the protocol for this particular study, is logMAR 1 or to logMAR 2.6, which equates to 2,200 vision on the upper end to 28,000 vision on the lower end. 28,000 vision, for some of you who may not know it, is something similar to counting finger vision at close to face. So again, we don't want to include patients with no perception of light or bare light perception, but we do have a significant range for patients to be included in this study. An important part of the study is we want to make sure that there are enough retinal ganglion cells and nerve fiber layer in the retina to be seen, and obviously this is examined by an OCT test. And what we do is we have the OCT sent to an independent masked reading center who will be reading that OCT and telling us whether the potential patient has any enough of retinal ganglion cells or nerve fiber layer to be part of the eligibility criteria. Also importantly, from an exclusion criteria perspective, if the patient has any other condition that precludes evaluation of the study outcomes, as per the PI, we would not be able to include that patient into the study as well. But these are some of the criteria for the study. Obviously, there's a whole range of inclusion and exclusion criteria that are defined in the protocol, but those are usually the routine ones that we see in pretty much all clinical trials. One important aspect of this study that has not been done previously in any of the studies is the Vision Research and Assessment Institute, that we have taken into consideration for the study. Evaluation of visual acuity in patients, starting from baseline testing all the way to eligibility testing after the treatment, will be done at the RAY, or the Vision Research and Assessment Institute in Irvine, California. The reason why we do this is because evaluation of visual acuity it requires time, it requires patience, especially in patients with vision ranging from severe to profound vision loss. It is a highly specialized endeavor, requires trained and experienced experts who have the time and patience, like I said, to conduct these tests. Clinical sites, just by the nature of the fact that they have so many patients coming in, are not able to do this test as well. So that's the reason why we have RAY as an important part of this study. So initial screening and safety visits will be done at the clinical site, but any of the efficacy testing after the treatment, patients will have to keep coming in to Ray in Irvine, California. And we do have a concierge service that will be working with patients to bring them over, especially if you live far away, if you live out of state of California or Houston, for example. You will be flown into the site and then also into Irvine, California, to get the assessments done at Ray. In regards to the Ray, I will also talk a little bit more later in the study, but later in the slide. But in regards to RTX-015, currently we have an ongoing study for retinitis pigmentosa, which has recruited about 7 patients so far. And what we have seen in the study is excellent safety in those patients with no issues in regards to inflammation on any other challenges that we have seen with other studies. So as I was mentioning earlier, I'm going to go a little detail into the RAY part. The RAY, or the Vision Research and Assessment Institute, is an important part for this study. But just to give a little more breadth about what exactly it is, it's led by experienced people who are experts in low vision and who also have validation of endpoints expertise in that particular area. What we have benefits from this institute is that we allow standardization of all the testing. So one center, everything is being tested. So there's no retest variability that comes into play when patients come into clinical sites. Also, in terms of recruitment, retention, and phenotyping of patients, it's an important aspect at Ray. Also, in addition, the center is involved in endpoint validation and development of endpoints, which potentially will be useful in this and future clinical trials. An important thing is there is an ongoing observational study at Ray, which is looking at all patients with IRDs or inherited retinal degenerations. So if you have, or if you know of any patient who has an IRD disease, feel free to contact Ray in terms of coming in, getting tested. I have the contact information on the next slide. And here we go. So there are, you can always contact Ray on the phone number or email given. And in regards to choroideremia, if you have any questions, again, you can ask, call up Ray or Ray Therapeutics on the phone numbers given here, or you can contact via email on the 2 emails. So on behalf, and this is my last slide, so on behalf of Ray Therapeutics, I would like to thank Choroideremia Research Foundation for this invitation to speak with you, and thank you all for this conversation. Happy to take any questions. Thank you.
And do we have questions? Thank you so much. This was great. And thank you, Rebecca, for staying on. I think Rebecca has a little bit of a time crunch. So if it's okay with the 2 of you, can I just go ahead and start posing the questions, and whomever would like to answer?
Sure.
All right, start with the Q&A. There have been a couple questions on both the chat and the Q&A. Can a female manifesting carrier be included in this study, or a female not necessarily manifesting eye loss or vision loss? Can they also be included?
So for now, most likely for this, like I said, this is a phase 1 study. So I would probably, say that we would probably not include patients at this female patients at this point, but probably for the phase 3 study that will ultimately go, the registration study, we should be able to include patients then, female patients then. But not in this study. We're only looking for 6 patients at this time, so we just want to make sure that we do it the way it's supposed to be defined in the protocol.
Does the treated cell function more like a rod or a cone?
That's a good question, and again, it's not been well defined as such, whether it's a rod or cone. I'm sure the person who asked this question knows about the differentiation in terms of what the cone and the rod does. So again, to mention in terms of what has been done previously in terms of animal trials and all that, there is perception of some color that potentially may come into play. So it might be working as a cone in some instances.
These two are very similar in nature. Will European citizens be eligible for this trial? And then I live in Scotland. Is there any scope for global expansion of this testing and potential treatment?
Again, a good question, and thank you for interest in this study. For this particular study, phase 1, we would probably not be able to include patients from outside the US just because of logistical reasons and the amount of flying that you will have to do to come in and get tested. But definitely, as we move on to phase 3 study for registrational and approval purposes, it'll be a global study. So you will be part of the study, depending obviously on eligibility.
That's exciting. Here is a particular question. What is the y-axis CS value units on those graphs?
I will have Dr. Kamat take care.
So that's basically the height of this curve. So let me go back a little bit. Contrast sensitivity is, if we just use one type of chart that's kind of been in use before, it's only going to measure one aspect or one size of the target, meaning the letters are one particular size. But contrast sensitivity is something that is across all sizes of objects. So imagine that you're looking at a little white pill on a gray background. That's contrast measurement of a very small target. But let's say you're also you're stepping off a curb and the curb is rather large, but it's light gray and the street is dark. That's a different type of contrast with a larger object. So our contrast sensitivity curve in vision should take into account large objects all the way to very small ones. And that's really the most complete way to look at it. And so when we're looking at the height of that curve, that tells us how close we are to kind of normal or non-impaired vision, which has a certain known peak to it, and then a certain look to the curve. But in low vision or visual impairment, that peak may be very suppressed, meaning you're only going to be able to see things that are really high black and white contrast and no grays at all. And then maybe you can only see large targets like the curbs or a big doorway that's black against white, because if you have impaired detail vision. And now we've got a really lopped off curve. Does that answer that? Well, it's hard to communicate, I think.
What's the process or eligibility to be considered for the Envision study?
Good question. Obviously, there are defined eligibility criteria for patients to be part of the study. As I mentioned, vision is one of the important part. Obviously, before that, more importantly, genetic testing has to confirm that the patient has choroideremia. And if you've not undergone a genetic test done before, like I said in my talk, we will be able to do the testing at the site and confirm the fact that the disease is choroideremia before we enroll the patient. So there are other defined eligibility criteria as well as exclusion criteria that is defined in the protocol. What I would suggest you do is, if you have questions about this, you're free to call Ray or Ray Therapeutics or Ray in Irvine, and talk to the people who will pick up the phone, and we will be able to provide you more, significant detail about the study at that point in regards to, individual perception about the study.
Another, excuse me, another person asks, have you dosed any patients with RP yet?
Yes, we have. So we have a total of 7 patients at this point of time being followed up as part of the study, same RTX 015. And so, like I said in one of the slides, what we're looking for is obviously safety. And so we have seen so far excellent safety in these patients. We are also testing them for efficacy as part of the study, and we'll be coming out with the details of that soon enough.
Does Ray Therapeutics pay for genetic testing to confirm eligibility, or does the patient need to be?
So again, good question. We would, if you are seen by the PI at the site, and the PI feels that you may have the choroideremia, or you've already been diagnosed as choroideremia, then we will be paying, the sponsor will take care of the testing charges.
I'm going to jump over to the other one, excuse me. Eligibility has been covered, so updating my question, where do we send genetic tests, etc., for consideration? And I think we're going to get some more of these in the chat. What is the best way for people to express their interest and get signed up? Or
Again, this is, thank you for the interest from the person who asked this question, and from others as well. I think it's important for us to, as I mentioned in this in the last slide, which is the 2 places that you can contact, which is Ray Therapeutics phone number or email, or Ray, which is the Vision Research and Assessment Institute. There are people who are aware of the study details, and they would be able to answer any questions that you have. And again, you can also contact any of the 3 sites if you wish. There are people there as well who will be able to answer questions, the clinical coordinators there.
all right, some are redundant, so I'm trying to go through them. In a previous intravitreal gene therapy trial, there was significant inflammation, and you did say something to this, Raj. Has Ray considered this complication?
As part of any gene therapy study, that stays in our mind when we design a study, or when we are, obviously enrolling patients with study. That's an important parameter that we have to keep in mind. Obviously, some studies have failed, or some studies have shown significant issues with information in the study. One of the things we are doing in our study, which is different, is number one, the vector that we are using has been proven to be less inflammatory or no inflammation at all in other studies previously. So again, we have enough evidence to showcase that this vector will not cause significant inflammation in patients. Secondly, we do dose patients with steroids, both oral and topical medications, topical as in eye drops, basically to control any inflammation that we potentially might be able to see in patients. Again, just to make sure that there is no inflammation happening in these patients. So far, like I said, with 7 patients that we've treated, we have not seen any inflammation in those patients. And obviously, if we would have seen some, we would have seen signs of that. We are not seeing any signs of inflammation in those patients at this time. So so far, it does look like, inflammation will not be a huge issue, or a significant issue for us in this study.
A follow-up to that question, fast forward to FDA approval, is there a hesitant to treat patients who have already undergone gene therapy in previous clinical trials? If so, why is that? And then do you have a projected timeline for completion of all phases in this clinical trial?
Excellent question. Obviously, one of the important things in people's mind, especially for those who have been part of previous studies, other gene therapy studies, is whether they can receive or be part of a study, or receive this injection as part of post-approval. We have to understand one thing very clearly, which is that FDA or any regulatory agency around the world, they define, which patients we will ultimately end up treating. So if they say that we are not able to treat patients who have previously received gene therapy, we will not be able to. But the point is that if there is, and also the reason why we don't do that is because the patient has been previously exposed to another vector, so we don't really know what may happen if we inject, let's say RTX-015 into those patients. So there is currently, we don't have enough evidence to showcase whether we should be able to treat or not. But that is one of the things that we keep in our minds all the time, because obviously patients have been exposed to other treatments before. And we hope that with some convincing data, we should be able to get the agencies to approve injections in those patients who have received previous gene therapy.
This is a specific question that might include several people on the call. Says person, I am located in California, diagnosed with late stage CHM. I have had cataract surgery and
Uveitis.
yes, sir. 24 years ago. Is that an eligibility criteria issue?
Based on what is being told, I don't think that that would be an issue. So we should be able to recruit the patient, but obviously the patient has to undergo all the testing and all the analysis before we take the patient up for study.
And just to clarify for the questions that are still coming in, to find out if you're eligible, contact Ray Therapeutics directly, correct?
Correct, absolutely.
And then is there a time frame expected for monitoring the phase 1 patients?
So we do have, the studies for 1 year. So once the patient gets the dose, patients will be followed up for a total of 1 year. And so there are defined time points for patients to come back to the clinical site, especially early on, because we want to make sure the patients, there is no safety issues. So patients come on a frequent basis early on, and then they start staggering over months. They also have to come back for efficacy testing to Ray in Irvine. So those are 4, at every 3-month timeline. So once they get those, the first visit will be 3 months after, and then subsequently 6, 9, and 12 months.
We have a lot more questions. I know, Rebecca, if you have to leave, please go.
I have to go. Yeah. Thank you.
Thank you so much for joining us.
You're welcome.
We'll go as quickly as we can. Rajan, when you have to leave, just let us know. Thanks for staying on with us. It says here, I was in a clinical trial at UW, but was just in a control group. Can I still be considered?
Excellent question again. And I think, based on the fact that you were on the control group, and if you're not injected, you could potentially be part of the study. But again, like I said earlier, we have to define, there are multiple different criteria that we have to take into consideration. So we will obviously have to analyze all those things before we, get you enrolled. Sure. But thank you for the interest.
Phase 1 is a very small controlled trial phase, but looking ahead in later phases, how many patients are you recruiting? Also, outside of Europe, will the study expand?
Good question in terms of, and obviously, like I said earlier, for a phase 3 study, we would be going global. So we will be recruiting patients from, additional sites over and beyond the US, I would imagine. Again, we are still in early phases, so nothing is defined at this point of time. What was the second question again? Sorry, I lost track there.
Do you know what? I already erased it. I'm sorry. I'm looking at the next ones that are coming. I apologize. Expanding into Europe. That was the one.
Yeah, I mean, we'll definitely have sites in Europe for sure. Yeah.
It's the second part.
And beyond.
Sorry about that. These two came in directly following one another. What are sort of pre-existing conditions would disqualify you from this study?
There's a long list in the protocol. I don't think I'll be able to read through it, but the primary thing is that what we're looking for is, example, patients with advanced glaucoma, patients with significant and current uveitis, meaning somebody has active inflammation in the eye. Somebody's got, let's say, a corneal issue that precludes us from looking into the eye and evaluating the patient. Anything that does not allow us to follow the patient up on a routine basis to showcase whether the patient is getting better or not. Those are the criteria that we would that would preclude the patient from being part of the study. And again, this is based on the PI at the site. So when the PI evaluates a patient, they look at all the aspects and make sure that they are okay with any patient with any disease. If it's stable, It's been around for a long time, as somebody else asked earlier about uveitis before. So if that uveitis patient is stable, has not had any inflammation in recent years, is not likely to, then maybe we could consider that patient. But again, it's on a case-by-case basis.
And I apologize if you answered this. I didn't track this question, but how many patients do you anticipate in phase 2 and phase 3?
Yeah, so the numbers will be definitely much more than what we are looking for in phase 1 study, of course. But somewhere in the range, again, I would not be, mentioning a finite number here, but somewhere in the range of about 100 to 200 patients, I would say, I would think, but definitely less than 200 patients.
And the timeline then would be if folks would reach out now, and you might not be considered for the first 6, but you could still get into the queue if indeed it progresses. Would that be the appropriate way?
That's the appropriate way. And also, as I mentioned, Ray is involved in a observational study of IRD patients. So it might help benefit the patient also if the patient comes into that observational study and be part of that study. So, we can monitor the patient as well. And then as we move on to phase 3 study, those patients can be, reached out to for being part of the phase 3 study.
Sure. For those like me who have enrolled but not been accepted into prior gene therapy studies, is there a much slow progression data available for over 10? Oh, excuse me. There is a much slow progression data available for over 10 years. Does that help for applicants to this trial?
I'm sorry, I'm not able to get the context here. Is it
Let me just read it one more time. It says, for those like me who have enrolled but not been accepted into prior gene therapy studies, there is a much more slow progression data now available for over 10 years. Does that help for applicants in this trial?
So in regards to the earlier part of the question, that you were enrolled in a study but were not part of the study, meaning you did not receive the treatment, again, based on criteria, if you fall within the eligibility criteria, you can be part of the study, number one. Number two, we do have, if I'm getting the question correctly, there is enough data for us to know the slow progression of the disease, or how the disease progresses over time based on natural history studies. So we do know some patients do progress slowly, some patients do not. And we also, there are people have actually figured out what kind of loss in vision patients have over time, based on the criteria of slow versus fast progression. I hope that answers the question.
Me too. I'm sure it did. Here's a very specific question. Can you talk about RTX 21021 and the differences and similarities with 015?
Oh, that's a question probably beyond my. I'm not a basic scientist. So again, to highlight, these are optogenetic treatments. Both of them going to be intravitreal injections, and pretty much similar on that account. But I think in terms of significant, very basic science level question, I'm sorry, I will not be able to answer that question because I'm not an expert in that space. Apologies.
If all goes super well west, would you expect to be what would you expect to be the time to market approximately?
That's a $1 million question. More than $1 million. If everything goes well, studies obviously have to progress well, and we have to, showcase improvement as defined and as required by the agencies. We do hope that we will be able to start phase 3 study based on the data that we receive from this study, hopefully very soon. And it usually takes, again, depending on the fact that, what numbers we are looking at in terms of recruiting for a phase 3 study, and the definition of eligibility criteria. If we are able to recruit patients well in time, we should be able to, get to the regulatory agencies. Obviously, there's a process, and once all that is taken into account, you get an approval. Again, if I'm looking into the crystal ball here and trying to answer that question, I'm looking at a few years from now. How many years? I think it's very difficult to say, but we're looking at somewhere in the range of 2 to 3 years.
If you're working with a specialist such as someone from Duke, should the patient consult Ray or should the specialist if you have somebody that's working on your behalf?
Good question. So it is important for you to consult with your, the provider that you've been to previously in regards to any, disease information that you wish to want to know. Um, Usually, what happens is most of the specialists in this particular space, for example, choroideremia specialists, they would be or they should be aware of this study. We also, as part of the community, we do talk to these people during conferences and stuff like that. So they are aware, and so they talk to the patients as well. But since you have a direct approach to Ray Therapeutics and Ray in Irvine, you feel free to call up the number or email any of the two emails that are provided, and we should be able to respond to those questions.
Corey wanted me to chime in and said, tell people to go to clinicaltrials.gov to see the listing of inclusion and exclusion criteria. Thank you,
Absolutely.
Corey.
That's an excellent point. Thank you.
Uh, Here's another quick question. Is the injection subretinal? And if so, is it localized to the area of remaining perceived eyesight like fovea or in an outside area?
Good question. As I mentioned in my talk, we are not doing subretinal injection. We are doing an intraretinal injection. Uh, Basically, the idea behind intraretinal injection is, it because it goes into the eye, it is able to spread around and be able to reach as many retinal ganglion cells as possible, making them open to optogenetics activity, and then be able to, be able to pass on that information onto the brain. So we do intraretinal injection, which is more safer, less cumbersome because you do not need to go into the operating room, do not need to have undergo specialized and complex surgery. Intravenous injections, as I'm sure most of you know, are given hundreds of thousands of times every day in the clinics around the world. It's a simple procedure, takes a few minutes, and patient can go home. So it's not a huge treatment burden for patients to get an intravenous injection. And as I referenced, uh, most likely this will be a one injection and one injection and that's it. So again, not a significant burden on the patient.
Let me just ask real quickly. We still have 75 people that are watching, and there are more questions. Do you still have time?
I have time, yes.
Okay. Okay, we'll keep on maybe for a few more minutes here. How collaborative is the approach towards the FDA in changing endpoints of all of those working on CHM treatments? Don't we know it? Is there one united voice on what is needed? Excellent question.
That's an excellent question. Yeah, I think we do definitely require what the questioner has asked here, that we do need a one voice approach to make things happen, and that's why I think working with Choroideremia Research Foundation is an important aspect of that. Um, FDA is open to suggestions, which is a good thing. Obviously they need data in terms of convincing them to change the approach from what they currently have. But good part of the process is that if we go with enough data, we go with a lot many voices, advocacy aspects as well. There might be hope for change. Again, we also have to understand the regulatory um, thought process. They want to make sure that whatever is being offered to patients as treatment is safe, primarily, first thing, but also efficacious. So they also have a hurdle to cross on their side to make sure that what the patients receive is good enough, both safety-wise as well as efficacy-wise. And that's the reason why they have set some norms. But like I said, if they have enough evidence to go and make a change in terms of endpoints, they should be able to do it. And all the work that we are doing at Ray is actually headed to in that direction. So we are doing endpoint validation, potentially new endpoints, which will ultimately hope change the way the treatments get approved.
Mhm, mhm. This is a different scope question. Like in the Nightstar and Oxford trial, when getting in later trial phases, is a pharmaceutical needed to invest in the further rollout? And thus, will a pharmaceutical be more interested in control what the endpoint and measurements will be for phase 2 and phase 3? We saw in Oxford's study that when the pharmaceutical came in, the 3-line chart metric was included, although specialists objected. What's your view on including pharmaceutical, and what is your view how much influence they have?
I presume pharmaceutical means the company that is involved in the study. And so I'm representing Ray Therapeutics, and we are a company as a sponsor of the study. Obviously, it is in our interest to make sure that what we design as part of the study and the criteria that we include in that particular design are tasked to make sure that it's the easiest way in terms of an approval. We don't want to make it too complex for patients or for the sites, and we also want to make sure that we follow regulatory advice. And that's the reason why we have ongoing conversations with agencies around the world. Again, it depends on acceptance from the regulatory agencies, whether they're open to suggestions. And as referenced, there were issues with the 3-line improvement criteria of endpoint for the studies done previously that failed. And we are hoping that we can convince the agencies to try and narrow it down a little bit or reduce it down, the high hurdle. And just to mention as a reference, the 3-line improvement that has been set by the FDA is actually not, it's very difficult to achieve in a patient with choroideremia or other inherited retinal degenerative diseases. That was meant for patients with macular degeneration and diabetic macular edema, where injections do show benefit in terms of improvement. So again, a little high bar for us to make sure that we have enough data to kind of get the agencies to approve a change in endpoint. But we are working towards it. So hopefully soon something will change.
If a person has AFIB, would that be an issue for inclusion? AFIB, I'm sorry, I don't know.
I am unsure of what that means.
Maybe it's a atrial fib. That's, I bet.
Oh, okay. Oh, yeah.
I bet. Not
Yeah,
sure, but she
I
did.
I'm a ophthalmologist, so sometimes these things
Cardiac.
take me by surprise. Uh, um, Again, if you're stable and if it's, I presume if the internist or somebody who's seen you and are, believes that, stability is the patient is stable enough with no issues, I think it should be not an issue for you to be a part of the study. But maybe you might be able to respond to that. What do you think? Let me throw the question at you.
I would think you have to go with your doctor's opinions and go through all those medical checks first.
Yeah,
I think that's kind of
absolutely. Absolutely. We need to get an opinion for sure.
We have a few more. I am a I'm sorry if I'm mispronouncing. I am a monocular, M-O-N-O,
Mhm.
monocular. So
Yep.
I'm sure I won't be able to get treatment. So just a question for everyone to smile. Doctors, do you agree CHM patients are usually more good looking? Stop. I shouldn't have. Stop. That was an inside joke. Okay. Yes. Okay. I'm sorry. Hey, this is.
But sorry to interject, but the monoocular part of the question could be answered. So if somebody is one-eyed, basically meaning that the other eye does not have vision, does not have visual perception, no light perception. Generally, those patients are not taken into studies, primarily because you don't want to, it's an investigative process, so you would rather not have any issue in a patient with one eye, one functional eye. But again, we would leave this to the PI of the site to define and feel whether that patient can be eligible for the study or not.
This isn't a question, but someone says, thank you very much. You are giving us hope for a future of our site.
Thank you. Thank you so much.
Would patients with, oh, here we go. If you had prior gene therapy treatments, could you be considered to enroll in a phase 2 or phase 3?
Again, we've not defined that as yet. Also, as I referenced earlier, we do have to, showcase a study design with the regulatory agency, and they have to approve of it. So if we do include that, and if they say no, we may not be able to. But again, this is a point for discussion for us internally, and we should hopefully be able to answer this question in time to come.
just for folks to remember, the CRF will, when we send out this in the video for this recording, we certainly will provide, and it will be on one of the slides, the email and phone numbers to reach out to the team. We'll make sure that's included. All right, we just have a few more left. Hey, you know what? We got through them all. That's it. Wow. Well, clearly, I'll tell you what, for the 67 people that are on, this is the most highly attended webinar we've ever done. So we're just really grateful, as Elizabeth said, we're just very grateful that you are taking time and continuing research, something that is so very important to us. As some people have alluded, we have gone through gene therapy trials and clinical, and it's been challenging. It's wonderful, but it continues to be a journey. So we are looking forward to are you attending the conference, or is someone else from your organization coming?
I'll be coming along with a few other people, yes. So
Awesome. That's a great segue. So everyone, you should be registering now. Early bird rates still for the CRF conference in Minneapolis, Minnesota, June 26th through the 28th. You can go to the website and sign up there. Please do so. We'll be having them here. Ray will be there, as well as others. And also, just if you're still on board, we are having a family chat tomorrow about technology for younger CHMers. So if you would like to join that discussion, there was an email that went out today. You can click on the link and we can get you signed up. Well, thank you, sir. Thank you for taking this extended time with us. We really appreciate it, everything that you and your team are doing.
thank you so much for this opportunity again, and thank you to everyone who's, I presume that there are people from Europe as well who logged in, so which is great to hear. As I mentioned earlier, we are working to try to make a difference, and hope that we'll be able to continue working in this space and make a difference to patients with choroideremia. So thank you again.
Oh, you bet. All the best of luck to everybody. Thanks, everyone. Have a wonderful day, and we'll see you next time. Thank you.
Thanks so
Bye-bye.
much. Bye then.
Thanks. See ya.